So hello everyone, we are going to study here chapter 6, that is in unit 2, the anatomy of flowering plants. In this we are going to cover these two topics, the tissue system and anatomy of dicotyledonous and monocotyledonous plants. So let's start. You can very easily see the structural similarities and variation in the external morphology of the larger living organisms, both plants and animals. Similarly, if we were to study the internal structures, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. A study of the internal structure of plant is called anatomy. Plants have cells as the basic units. Cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in a plant show differences in their internal structures. Within angiosperm, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptation to diverse environments. The tissue system. We were discussing types of tissues based on the types of cell present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue system. These are epidermal tissue system, ground or fundamental tissue system, and the vascular or conducting tissue system. Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gases exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose the stomatal pore. In grasses, the guard cells are dumbbell shaped. The outer wall of guard cells are thin and the inner walls toward the stomatal pore are highly thickened. The guard cell possesses chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of guard cell become specialized in their shape and size are known as subsidiary cells. The stomatal aperture guard cells and surrounding subsidiary cells are together called stomatal apparatus. The cells of the epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branch or unbranched and soft or stiff. They may be even secretory. The trichomes help in preventing water loss due to transpiration. The ground tissue system. All tissues except epidermis and vascular bundle constitute the ground tissues. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchyma cells are usually present in cortex, pericycle, pith, and medullary rays in the primary stems and roots, and also in the primary stems and roots. In leaves, the ground tissues consist of thin-walled chloroplast containing cells and is called mesophyll. Here is the vascular tissue system that contains the xylem and phloem. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues, and hence are called open vascular bundles. In the monocotyledon, the vascular bundle have no cambium present in them. Hence, since they do not form secondary tissues, they are referred as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In cone joint type of vascular bundle, the xylem and phloem are jointly situated along the same radius of vascular bundle. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stems, and leaves, it is convenient to study the transverse section of the mature zones of these organs. Dicotyledonous root. Look at figure 6.3a. This is the figure 6.3a. It shows a section of transverse section of sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. One of the cells of epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of cortex is called endodermis. It comprises of single layer barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material submarine in form of Casparian strips. Next to the endospermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between xylem and phloem are called conjunctive tissues. They are usually 2 or 4 xylem and phloem patches. Later, a cambium ring develops between xylem and phloem. All tissues on the inner side of endodermis, such as pericycle, vascular bundles, or pith, constitute the still monocotyledonous roots. The anatomy of monocot root is similar to dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. Dicotyledonous system. The transverse section of typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may be a trichomes and few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, when provide mechanical strength to the young stem. Cortical layers between below hypodermis consist of rounded thin-walled parenchymatous cells with the conspicuous intercellular spaces. The innermost layer of the cortex is called endodermis. The cells of endodermis are rich in starch grains, and layer is also referred to as starch sheath. Pericycle is present on inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundle, there are few layers of radially placed parenchymatous cells which constitute medullary layer. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is a characteristic of dicot stem. Each vascular bundle is conjoint, open, and with endosperm protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central position of the stem constitute the pith. Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located one. The phloem parenchyma is absent and water-containing cavities are present with vascular bundles. Dorsiventral leaf. The vertical dorsiventral means dicotyledonous leaf. The vertical section of the dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll, And vascular system, the epidermis, which covers both the upper surface adaxial epidermis and lower surface abaxial epidermis of the leaf, has conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called mesophyll. Mesophyll, which uses chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cells, palisade parenchyma and the spongy parenchyma. The axially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extend to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and the midrib. The size of vascular bundles are dependent on the size of veins. The veins vary in the thickness and in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick walled bundle sheath cells. Look at the figure 6.5a. 6.5a. This. Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that dorsiventral leaf in many ways. It shows the following characteristics and differences. In an isobilateral leaf, the stomata are present on both surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stretch, they make the leaves curl inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles, except in main veins, as seen in the vertical section of the leaves. Summary. Anatomically, a plant is made up of different kinds of tissues. The plant tissues are broadly classified into meristematic and permanent tissues. Assimilation of food and its storage, transportation of water, minerals, and photosynthesis and mechanical support are the main function of tissues. There are three types of tissue system: epidermal, ground, and vascular. The epidermal tissue are made up of epidermal cells, stomata, and the epidermal appendages. The ground tissue system forms the main bulk of the plant, divided into three zones: cortex, perishal, and pith. The vascular tissue form xylem, phloem on the basis of presence of cambium, location of xylem and phloem. The vascular bundles are of different types. The vascular bundles form conducting tissues and translocate water, minerals, and food materials. Monocotyledonous and dicotyledonous plants show marked variation in their internal structure that differ in type, number, and location of vascular bundles. The secondary growth occurs in the most of dicotyledonous roots and stems. Our chapter is finished. So thanks.